Thank you very much for coming. And Rachel, would you like to introduce Kate as normal?
Sure. Well, yes, as Selena said, thank you all for tuning in. It is so wonderful to see everyone's faces in 2026, back at our monthly net chatter. And to kick off this year, we are so excited for Dr. Kate Lines to be here and give us an update on the 2025 Net Models meeting, which occurred back in September of 2025. So we're really excited to see what updates she has in store for us. So without further ado, Kate.
Perfect. I will now try sharing my screen, which is always the fun bit. So please tell me whether you can see things. I will put it on presentation mode. So can you guys see that okay?
Uh-oh, when you hit presentation mode, it went away.
Oh, that's fun. Okay. Let's
I
well, I
haven't
could
seen that happen before.
well, let's try that again and see what happens. If not, I tell you what, let me just share my whole desktop and then hopefully that should bear with me
I
whilst I get
see
rid of all.
Poe in her new office.
I was going to say, let me, can you now see the
Hi.
screen?
Yes, Kate, that looks good.
We'll do it that way. It's a bit more convoluted, but we'll do it that way. And I can't see any of you now, but never mind. But no, I just thought that I was just saying that it'd be quite timely to go through this because we're planning on putting the white paper together very soon. And also, we've just had some confirmation on what's going to be happening on the Net Models meeting in 2026. So I thought I would kind of give you, for those that haven't maybe heard about it before, a general overview of what this Net Models meeting thing is about, and then the outcome of what happened this last time round. So basically, this all came about through the Net Models Consortium, which we started up quite a few years ago now. And the whole aim of it was to bring together researchers from across the world to improve the quality and access to preclinical net models, because I think it was becoming painfully obvious that we were working in sort of silos away from each other. And with the rarity of the samples, availability, and skill sets, that working together was going to be kind of the only way that this would happen. And this actually came about quite fortuitously, I think, with a conversation between me and Dawn at Endo one year. So it's quite nice to know that those conversations you have at these meetings actually can get you somewhere. But basically, the consortium was sort of subdivided initially into three groups. That was in vitro, ex vivo, and in vivo. So this list looking at different types of models. The first being kind of cells in dishes, that kind of thing. The second one is the sort of more 3D cultures that are a bit closer to sort of patient material, but they're still kind of in a dish. And then in vivo is all your sort of animal models. And we have kind of two people heading up each of those. So me and Dawn are currently in charge of the in vitro side. Poe, so hi Poe for being here as well to help out with some of this, and Talia are part of the ex vivo group, and then Ilaria in Switzerland and Rachel, again another one, hello, are currently in charge of the in vivo group. So we're trying to get sort of a transatlantic representation, and hopefully you can see we've got sort of Europe and the USA covered as part of this. So we actually held our first meeting in 2024 at the end of February. And that was kind of a pilot test run to see how it would go. It was actually partly organised by Bioscientifica, so it was a bit more sort of formalised. And we actually had 26 attendees come to that from different parts of the world. It was a 2 day meeting, and we had a couple of plenary talks, one by Raj Thacker and the other one by Jörg Schrader. On top of that, we had 8 researchers presenting their own work, with a real big spectrum of things discussed. So everything from 3D bioprinting of cells all the way through to zebrafish. And also things that were very novel were discussed, as well as improvements on sort of the current concepts, that sort of the mainstream concepts, for example, cell lines and organoids. But the big part of this was that we had a workshop to get everyone together to discuss the sort of limitations, barriers, and what our aspirations are for the future. And that then basically led into this white paper that we published. And part of the reason for doing this was that we kind of wanted it on paper to show what the challenges really are and how in our field we're kind of facing some that a lot of other fields aren't. So it was also to help us kind of put our names out there. So when we're applying for grants, etc., people have maybe a bit of an understanding as to why we're asking for certain things. So it could kind of be a reference guide. Um, so that was great, and the kind of the key things that came out of that. I mean, this paper's published. You can find it online on the Endocrine Oncology website. Um, but I think a lot of these things to this group will come as no surprise. That basically we said that the current state of models for doing preclinical work, so those cells, tissues, etc., that I was mentioning, are severely lacking and are desperately needed for us to be able to address the really important basic and translational questions that we need to help cure this disease, to help better treatments, management, biomarkers, etc. Um, and a lot of this is grounded in some real basic science because actually basic science in this field is quite lacking as well compared to a lot of other cancers and diseases. Um, we also kind of highlighted that yes, we have a number of models already, including cell lines that have been very well used, used to the best of their ability. But that is also narrowing our research a little bit because the diversity of these is quite limited. And we know that NETs are a very big group of cancers. So there, isn't just one tissue, one organ, one genetic mutation involved. It's really covering quite a large area, which I think is quite unique compared to most other diseases. And actually, the more information we find, the more this evolves, and actually, the more improved models we need. Um, Also, that we said that the development of organoids, so these are these little sort of mini tissues, is expanding, but some of them are much easier to establish than others. So, for example, trying to get a sample from the pancreas and to get that to grow has been a really big challenge for years. Again, obviously, many other places aren't facing those same problems, and there's lots of different reasonings behind that. Finally, mice are the most widely used in vivo model, but they're also very limited in their sort of genetics and the type of tumours they get. Quite a lot of them are heavily focused around the pancreas, ironically, considering the models are poor in the other areas. But again, they're not particularly well representative of the patients that you would see in the clinic and the tissues that you would get there. So clearly we already knew going into this is why we were organizing this meeting, that we had all these challenges. But it was quite nice for everyone to just kind of put it down and on paper and really get it in black and white, vocalize to kind of the rest of the scientific and medical community. Um, then we kind of delved a little bit more into why this might be happening and what we could do to sort of try and address it. So we've said one of the major issues is things like access to patient material, a lot of red tape, which means that it's very difficult to get ethical approval, etc., to share patient material. And that is actually a real burden when you're looking at sort of fairly rare diseases and material you don't get ready access to. Also, there's quite a lack of purchasable resources. So, of course, for things like breast cancer, there's a kind of more commercial element to it. So, it's worth their while with pharma companies having a, repository of things that people will buy because they're going to be frequently used. But of course, for things like neuroendocrine tumors, they're much less in demand in certain ways. So, there's not that incentive for them to do it. Um, Cost is always going to be a problem for everything. Um, Money generally, I think, in science is lacking across the board at the moment. And so you're vying for that money against other people who maybe can sell a better story that they can use, sort of, newsworthy is always a term that's sort of batted around. Um, Also, I sort of alluded to the availability of expertise and equipment. So, as I said, we were kind of siloed around the world. There weren't these big powerhouse groups in quite the same way, again, there are for other diseases. And kind of what comes hand in hand with that is the transparency of the work we're doing. So whether that be the specific methods or protocols, the sort of things we're doing day to day to verify that what we're using is correct, and that these models are actually representative of what we think they are, because we weren't talking to each other very well, to be honest. So that was kind of one of the things also we wanted to address. So the future aspirations of that meeting, we kind of said we wanted to bring people together because collaboration is definitely the key to this. We need to get better at sharing results and information. Where possible, we need to actually make the resources we have available away from people having to request it exactly through us. So that does mean depositing them in these sort of company data banks that they have that people can then go to and purchase things. We also really need to start thinking about collaborating with other disciplines. So by that, I mean, sort of engineers even. So really kind of expanding our repertoire of things that could be available to us. And then the really big thing was that we were saying we need these bigger sort of collaborative groups of people to come together to get funding applications between them to kind of help improve the sales pitch really and show that we do have a lot of resources and skills between us that we could utilize. So that was kind of the summary from the 2024 meeting. But thankfully, we then got on board with the NETChatter Foundation, who very kindly helped us with the 2025 meeting. And this had a few changes because we parted ways with Bioscientifica for a lot of reasons. But it meant that we could kind of mold this meeting to be a bit more, having reflected on the first one, a bit more how we wanted it to go, a bit more sort of collaborative. And, you know, the most important thing was being able to get people in the room. So this one, the one big thing we've got there is this one was free to attend. Yes, couldn't pay travel, but there was no registration cost, which was different to the first one because obviously Bioscientific are a company and they also wanted to make a small amount of money from it. But this one showed an expansion of the number of people that attended. Again, we had them from all around the world, and you can see a nice little photo there that was taken of the people that attended. Again, it was a 2-day meeting with a couple of plenary talks. But this time we had 18 researchers talking about their work that they're currently doing, and that ranged, I think, even greater in span than it did the first year. So everything from mathematical modelling through to using fly models, so Drosophila. And this again included sort of novel concepts and ideas that were in very early stages. Whereas I think the last meeting, it was more sort of well-established things, but this was a bit more sort of idea generating and including things like comparative oncology. So that is basically looking at other animals that get cancers, which I'll talk about again in a minute, but basically predominantly dogs and cats. And also extracellular vesicle detection. So one of the people that attended that I work here with, at Brooks with is actually an expert in extracellular vesicles. And these are really tiny little sort of they're kind of like little droplets that are released from the tumor cells into the circulation. And with the increase in technology, you can actually start detecting these. You can isolate them out of the blood of patients. And that opens up an opportunity to look at a number of different things, including you can look at DNA, so genetic changes, proteins, and even RNA to look at things like microRNA changes. So there's quite a lot of things here that were brought to the fore that I think we could utilize going forward for net research. But again, we also wanted to have this workshop to kind of have another discussion about where we are and why we think things are moving in certain ways, what the barriers still are, and actually whether there are any improvements from the last meeting, whether actually that meeting did lead into anything. Another really big update, which we were very pleased about, is that we actually added an additional subgroup. So in addition to the sort of very model-centric ones, we wanted to add one which was patient involvement in net research. And that was actually headed by Selena and John, who are both here. So you can correct me when I talk about things you said that are wrong. But also another thing that we were very proud of and very pleased we could do was that we wanted to kind of encourage the future of net research as well at this. So we were quite keen to award a prize to the best presentation given by a very early career sort of person. So we decided to go with a PhD student. And we called it the Simon Robinson Award for the best PhD student presentation. And as you can see there, there's Phoebe getting her little trophy for her presentation, which was really, really good. And I think we really do want to support the sort of early career people in this as well because they're the future and we need to get them involved and passionate about what they're doing so that they can, really keep going forward with this. Also, what was really nice is that Simon's daughter was there, who also was there to give the award. And hopefully, I don't need to explain to this group who Simon was, but obviously, he was the founder of the Net Cancer Foundation and Selena's husband. Yeah, it was really nice because all of us worked with him at the beginning on this. So it was really nice to kind of be able to honour him in this way as well. So we're really pleased that we're working together because I think, for the Net Cancer Foundation and for Simon and obviously Selena, this is really kind of what the whole point of the foundation was for. So it's really nice that we can kind of continue doing this and driving things forward. But then some kind of a bit more details about what happened at this meeting. So I said there were two plenary talks. So those of you that have worked in the net field for a while probably have heard of Jörg and Raj. They've been around a long time. Both very, very good at what they do. But I think what we also wanted to do was try and sort of diversify and bring in some sort of new blood into the situation. So the two plenary talks we had here, they're still very, very good, well-established researchers, but maybe not as sort of well known as the ones from last year. So Cynthia is actually a professor at King's College London, and her kind of main sort of research area is endocrine stem cells. So she's particularly actually interested in research in the pituitary and adrenal glands, which of course are both very relevant for neuroendocrine tumors. But she does a lot of kind of state of the art, I would say, techniques, looking at lots of different details of the biology of these different tissues, and in particular, these stem cells. So these are the cells that kind of are the cells that can maybe be the kind of root cause of a lot of these cancers that are harder to treat, that can evolve a bit better. So they're really important to look at, but are much fewer in number compared to the sort of established cancer cells. So kind of the line of thought is that if you don't kill those off, then the tumor can just keep coming back because they can just keep designing themselves into being different cancer cells going forward. So that's really kind of what her research is focused on, and she's doing all sorts of fancy things as well, using like chicken eggs as a model. So it was really good to have her there. Um, Florine as well was another one that has been doing some really good work recently. Actually attended the Net Models meeting the year before, but she's actually a small vet. It's not a small vet. She's a small animal veterinary surgeon. She's one of the leading cancer surgeons in the country for dog cancer. But what is interesting is that the more conversations we're having is obviously she treats dogs like patients and they get tumors that occur just naturally. They have very similar sort of environmental settings to people. So actually, they're proving to be a really interesting source of research to compare sort of human and canine, so dog diseases. And actually, there's a lot of parallels in the like treatments for both because I think like she was saying that a lot of the treatments they use to treat dogs actually are kind of extrapolated from human research. And of course, we can learn a lot from the studies they can do with the dog samples once they've got them that are maybe a bit more challenging in humans. So it was really interesting to get her kind of take on things. And she's doing some really good work there. And that's what the comparative oncology is that I was talking about. So it's comparing, obviously, sporadically occurring or genetically occurring cancers in dogs. She particularly is interested in insulinomas and sort of pancreas occurring and also adrenal tumors. So again, very much sort of in the endocrine and net space. So it was really good to have her there as well. But then the kind of key findings from this workshop, we kind of wanted to move forward a few stages from before, because I think the first one was very much like, where are we? Kind of stating things that we already knew, but just being a way of making them a bit more public. Whereas this time we wanted to delve into a few things in a bit more detail. So as I said, this time there were then four subgroups. So again, the in vitro, which I actually led the discussions about, ex vivo that was led by Poe, and then the in vivo that was led by Rachel, and then the new one, the patient involvement in net research, was led by Selena. And I'm very pleased that all of you are here. So all the things that I say, or if any questions come up, then we can have a little discussion about it together, which is really nice. Um, but the first three actually answer slightly different questions to the patient involvement group. So I'll go through those ones first, and then I'll go through the other ones a bit after. Um, but these were kind of the four key questions that they were asked to address. So the first one is, what developments have there been over the last year? That was mainly focused around sort of publications, collaborations, and grants. So that was kind of just literally since the last meeting. So not everything that had been sort of in the pipeline for a long time, but kind of immediate things that had been achieved. Just to kind of get a grip on how quickly we're moving forward with all of this stuff. The second one was how to facilitate collaborations, particularly from the different fields that I'd mentioned before. So, especially, I think everyone will say, with AI coming quite to the fore, that many of us are not experts in at all, things like that. And how can we work with them, maybe engineers or other scientific disciplines, to kind of help move all of this work forward? Um, and then one of the other big questions was how we can validate models to ensure that they are accurate and representative, but also are being used appropriately for the research questions being asked. Because I think that's a big thing, that especially people that are less established in this field, it's whether they're aware of some of the limitations in quite the same way and are using them for sort of the appropriate experiments. And then, of course, finally, also was kind of our future aspirations from this, although I'll address that all together after we've gone through the patient bit as well. So, what developments have there been? Well, actually, quite a few. So, I'm not going to list every publication that's come out because there have been a lot, but I just wanted to highlight a few of the things that were mentioned. So, apologies for how boring these slides are, because it's just a lot of words. But there are definitely a number of manuscripts that are currently in revision or in press on net organoids that are coming to the fore. I'm personally reviewing one at the moment. So there's about 4 or 5 groups that have all got these in the pipeline. So hopefully they should all be coming out in the next sort of few months or so. So there really is a lot of work going on in that organoid field sort of at the moment. One that was actually is already been presented quite a lot, which I wanted to highlight, is the genetically inducible Gepner organoid that was presented by varying different members of James Yao's team from ND Amson at different meetings, including NetRF, NANETS, eNets. So that's been talked about quite a lot, and also the Net Models meeting last year. So, there is a lot of work going on in that space at the moment. There have been a few new collaborations and grants that have been established, which is good as well. So I personally actually managed to get a small grant awarded last year with Florine, who was our speaker, and that was to study epigenetic mechanisms in dog versus human samples. And basically our hypothesis there is, I very much am interested in epigenetics. So that's sort of inheritable changes that don't affect your DNA sequence. So that can be lots of different things, from different chemical groups being added onto your DNA, through to changes in structure of the DNA in the cells, which can affect how different genes are expressed. And we think that dogs are much more representative of humans than things like mouse models or laboratory models would be. Mainly because they live alongside humans. So, of course, they get the same exposures to things, like chemicals or pollutants, etc., which can potentially affect this epigenetic state. Also, things like their diet, etc., can maybe not, can vary, and they have that sort of baseline that an animal being bred in a laboratory just wouldn't have, because it's obviously too much of a sort of sterile environment. We also have other collaborations. So, for example, I'm also working ironically with a woman stood behind me, who you might see, who's just leaving, working on Drosophila models. So those are the fly models to study sort of better biology of menin function and other genes that are associated with neuroendocrine tumors. So we're doing that here at Brooks. So we have a little bit of money to look at that. But we think actually there are some really good experts here that have been using flies for a long time to look at this. And I think that's a bit of a sort of an untapped area for us that although people started looking at that a number of years ago, I think the momentum was lost somewhere and it's not being used quite so much. But they're actually a really good tool for looking at changes in genetics and biological function. And I'm finding out some interesting things. For example, I didn't realize that the fly brain is actually its kind of endocrine gland and it secretes insulin from its brain and has a little gland attached to it that has all the other hormones it needs. But it's quite good because it's all sort of in one place. But it does have an endocrine system, which I don't think I realized that flies did have a proper endocrine system. So that's proving to be useful. Um, Also, the MD Anderson team, so that's again James Yao, who received a prestigious grant from the Cancer Prevention and Research Institute of Texas. And that's to kind of further develop those GEP-NET organoid models that I was kind of mentioning before. So it's really good that that extra grant is coming in. And also Iowa, mainly led by Dawn, have also received the NET Specialized Program of Research Excellence, so a SPORE from the NIH, to look at advanced novel therapeutic options for NET patients, and also to help build sort of NET research communities. So there is stuff that's happening, and I think we kind of need to be aware that all this is going on, and a lot of this is coming about because we're starting to work together. But what you'll notice is there that those collaborations are still quite isolated. So, for example, obviously the Drosophila models, it's me and the woman I share an office with. Um, Obviously, yeah, me and Florine, but that's sort of London compared to Oxford. So I think, there's still a little bit of work to do there on sort of developments, and hopefully that will organically happen over the years as research increases. So that leads nicely on to collaboration. So how do we foster these collaborations and what can we do? Um, so this is what we were saying. We need to kind of be better at blowing our own trumpets, I think. And we kind of collectively, a number of different people said this, that we need to be better at showing our progression and also showing how that is producing research opportunities for people in different fields. So I think actually, personally speaking, as a scientist, neuroendocrine tumors are fascinating. They are so different to so many other things that actually you have scope there to do some really, really good research that you wouldn't be able to do in other things. And I think we need to be better at kind of advertising that to the wider sort of research, scientific, medical sort of field. So they might be a little bit more interested and see those opportunities that maybe they're not at the moment. Um, but what comes hand in that, I think I personally know I do this. I do a lot of whinging because we complain about the models. We've got a lot. We're trying to do things. We complain we don't have a lot of funding. But probably what we really need to do is articulate some very clear problems, some very sort of specific, fairly small problems, show how these are unique to nets, and then see how we can get people to help with those. So I think at the moment, a lot of us make a lot of noise generally, but we don't have a very precise problem that we are wanting people to help us address. That's, not everybody is the same, but I think publicly, that's what we were saying, that it'd be really nice to kind of articulate some of those maybe a bit better. Um, I mean, in an ideal world, it would be spectacular to develop a fully funded Net Models Consortium or a virtual centre where we could work together officially to do this. I mean, that would take a lot of time, effort, and money probably to do that. But there are ways that maybe we could help with that in the interim. So one thing that was definitely mentioned is possibly we need a better virtual platform or a website that's really bringing everyone together and kind of a public facing way that shows what the problems are in their sort of net models. And then hopefully that acts as kind of almost like a research place for people to go to in other fields who might want to get involved with us or might want to look at opportunities. And I think the only way we're going to get forward is we're going to have to continue multidisciplinary funding applications. So we need to, when we're writing our own grants, we need to be thinking about who we can get on board, who we can approach, and then slowly but surely, hopefully, we can sort of chip away at these other funding bodies, at these other experts, and they can see kind of what the benefits would be of working with us and that funding is there and available. Um, so then looking at the sort of validation and assessing to check that these models are being used for the correct purposes, really. I mean, this is always going to be a challenge. I think one thing that's been mentioned again quite a lot is that we probably need to publish guidelines to help with this standardization. So, for example, a colleague of mine, Ryan, who's head of the Extracellular Vesicles Society, they actually did this together and they published like a gold standard methodology basically to say, how you should be doing things and what things you should be doing for verification. And I think maybe not, obviously this is still quite a broad field, but maybe we need to start thinking about putting some things like that out to say, if you are using a cell line, what should you be doing when you're publishing it? What sort of authentication should you be doing? What sort of verifications of expression of key markers, etc. Because I think a lot of people maybe aren't aware of what should be happening and the sort of risks of not doing that. Because there is still even fairly high profile people that have had to have papers altered or retracted because of errors that were discovered later. And it's not maliciously that people do that. It's just that if you don't realize, then it's easy to make small mistakes, which can then be quite problematic for people trusting the rest of your studies when actually they're all been done very well, but you don't want to have that element of doubt. Um, so I think also for us within the sort of consortium or within this immediate research field, I think we also have to kind of set the standard. Um, so I think we also have to make sure that we are doing these things and that it's being published in all the papers we do and that we're stating it. And also if we're reviewing papers that we maybe ask for it. Because again, as I said, I don't think people are like missing it out on purpose, but it can get lost in word counts and things like that. I think it was also mentioned that it might be really good to do some sort of systematic review of kind of the data that already exists and what sort of validation has been used already and kind of what that's meant for the publications and the research coming out of it. Because it might be that actually the validation isn't that important at all and that generally everything that's been put out is really good, which hopefully is the case. But I think that, there might still be some merit in going back and looking at that. Another big thing really is to set up a database of the models, validation and access to those models. So again, this is almost like a lookup table where if people are using a model, they can look it up and say, oh, okay, what validation have people done before? What do I expect this to look like? But also, if I need to go back and if I think mine's wrong, where can I purchase? Who can I approach to get sort of information on this? Just so it makes it a bit easier. Because at the moment, I think you're sort of in the lap of Google and, knowing people rather than it being a bit more sort of easy to look up. Finally, I think, one thing is, can we use AI? I think a lot of people are quite scared of it at the moment, but is there any way that we can use AI to help with this? Um, Certainly not my area of expertise, but there is potential that some sort of AI or modeling could also help and also to ease some of the workload of maybe some of these things we've mentioned, because the reality is a lot of these are quite labor intensive. And I don't know, really how much time people have got available to do things like this. So that's kind of the summary of what came out of the sort of four subgroups. Now, obviously, I've done that very generally. There are obviously more specific points that fall within each of the categories that relate to like cell lines, organoids, etc. But I'd be here sort of all evening if I presented all of them. But obviously, we are currently drafting that white paper that will be coming out in endocrine oncology, hopefully later this year. So all of the details will obviously be in that. But I also wanted to give some time to the patient involvement group because that was the new one. And I said we gave them slightly different questions because this is a really important part that we can't get these models, we can't make them unless patients are willing to engage and also give their samples. And I think that's been a bit of a challenge sometime as well. But also patients can help advocate and really push kind of the research as well if we find the right way and outlet to do that. Um, so the four questions that they were asking was, what does patient participation actually mean? How do we get patients to consent more easily? What are the current barriers? What is the current state of advocacy or patient support? And again, what are the future aspirations? Um, so generally, I think it was concluded that participation could cover a lot of different areas. So that's everything from trials, so like clinical trials, natural history studies. So a lot of that might just be looking back at sort of records. Obviously, then tissue collection. So are you going to donate, tissues if you have to have operations, etc. And the last one is sort of responding to surveys or questionnaires that clinicians might give out about, some of those could be sort of like welfare related, mental health, some of the more detailed about your sort of clinical history. Um, so that's kind of research. But there's also the other side of it, which is community participation. So, of course, that's are patients joining these advocacy groups? Are they interested in joining? Are they not? Um, and if they do join them, are they actually participating in the things that are going on? Because it might be that they look at websites, they might get leaflets, for example, ask for advice if they need it. But are they actually participating in these events that are, whether it be online or in-person meetings or, there's lots of different sort of social engagements and things that are done. So it's like, is that element covered as well? So then the sort of big one was consent, because not every patient wants to consent for their samples or their information, etc., being used. And there might be a number of different reasons for that. And again, obviously, I'm not the expert. We have some patients here, which I'm sure know far better than I do. But this is kind of the conclusion of what was discussed at the meeting, which did include patients, I might add, and some clinicians. But I think one of the top things that was mentioned is you need an inviting environment to want to consent and discuss being part of a research study. Because if you're not feeling comfortable in that, you're certainly not going to want to be involved. And again, maybe coming slightly in that comes with better education. I think, a lot of clinicians particularly are busy. It might be that you get sort of given a leaflet, but actually that kind of, real education on research and the purpose and need for it. It might even be, for example, there's a scientist who's doing the research and they're not even the person you would ever see. You're going to see the person who's, your clinician that you're meeting regularly, or your surgeon or whatever, but you might not ever actually get to see the person that's doing the proper research behind the scenes. Um, so I think there are ways we could better maybe sort of get across the benefits of this, and that is not only obviously for the future of research, but actually there can be direct benefits to the patient themselves at the time. And it's whether all of that is getting across to help those patients make a really informed decision on or whether they want to be involved or not. Um, there is also this burden for multiple consents. So I think, for example, if multiple different hospitals or centres are used, if you change your mind and want different samples later, you can often be in a situation where patients being asked to sign consent forms quite a lot. And I know in the UK particularly, those consent forms can be really long, and I assume quite intimidating. So it might be that you don't really want to be doing that repeatedly, especially if every time you go to a different hospital or visit, they're asking you to sign a different form. So I suppose it's kind of about streamlining that, which sometimes, unfortunately, because it's not our decision a lot of the time for that. Um, I think this has kind of been also mentioned for science, but I think it's also very important for patients, and that's the better feedback of the results. So the fact that we are doing things with these samples, and although we can't get on the, national news website to say we've cured something, actually we're chipping away and we're doing some things that are really actually quite impressive and are really helping to move things forward. And it's whether we're good enough at kind of sharing that, because in the world of research, stuff can get lost in the noise quite easily. So it's whether we can find outlets to really feedback properly the results that aren't just these snippets, sort of patient meetings that maybe not everyone can access or go to. Um, there's also this need for maybe trying to improve convenience of being involved in studies. So, for example, things that might involve multiple visits or having to turn up just to fill in a questionnaire on a piece of paper. Maybe patients are going to not go because it's inconvenient rather than them not wanting to be interested. So I think discussions need to be made on how we can sort of cover that. And alongside that, also maybe improve the language to be more patient friendly. That doesn't mean make it basic, so that you don't understand, but I think also sometimes we can be a bit cold in the language we use because obviously we're coming at it from a sort of a different angle, and whether maybe conversations need to be had about that. Also, a big thing is ensuring clinicians have knowledge of trials and opportunities that are ongoing, because I think sometimes patients go to the clinicians rather than the other way around, saying they've heard on the grapevine. So it's whether there's an outlet way we can sort of make sure that clinicians are made aware of that, especially in the more peripheral centres. And of course, the big one to implement all of these is funding, which again happens across the board. Um, but also the current state of advocacy. So I think there are many organisations, which is good and bad. I think different countries have different organisations, and obviously we want there to be the support. But maybe they also, to some extent, need to be better at working together to ensure that patients don't have to kind of choose which one they want to use. Um, and kind of one of the barriers, again, with accessing this could be that clinicians and providers might not actually be aware of all the different support groups. So maybe along the lines of trials that might be available, maybe they need to be more made more aware of all the patient support that is out there, because someone has to fight against this misinformation, which is getting more and more rife on social media, and especially with AI coming to the fore, you can search things and get a lot of stuff that is completely wrong. And I think that really sort of needs to be addressed. And obviously, the formal sort of patient support groups are one of the best ways to do that. Um, and maybe also looking at that is sort of leveraging sort of more centralized international groups like INCA and Healing Net in the US, for examples, to help educate the providers or the clinicians to kind of help support with some of those things. Um, so then the future aspirations, I've kind of gone through as I've said this, but just to kind of lay them out properly, is definitely collaboration and these bigger sort of funded team projects is what we're aiming for, and kind of maybe thinking a bit more outside the box. So looking at like, computational modeling, bioengineering, AI, all of that sort of stuff is probably going to be important, and growing these partnerships, not only with sort of clinicians and scientists, but also across patients and patient support groups, and having these sort of like more long-term partnerships rather than just these short funded collaborations that are then going to fall apart if the money disappears. And as part of that, it's not only sort of research, but sharing samples, reagents, knowledge across multiple different institutes as well, if possible, to really sort of help drive these things forward. Um, as I've mentioned a lot, improving communication, both with experts and patients. So maybe in the sense of setting up some sort of online platform, even if to start with, it's just a website. And either through that or elsewhere, we could start up some sort of working groups that are working in each of these areas, again, to kind of be that kind of long-term connection that people can get together and have an input and ask questions if they need help with things. And as mentioned, setting up a database of existing models that will be helpful for so many different reasons. So that's kind of all the information on this last meeting we had. So I just wanted to finish by saying that it is now official, it is confirmed, and details will be coming out shortly. But the Net Models 2026 is actually going to be taking place in the USA, not in the UK, like it has done for the last 2 years. It's going to be a bit more of a streamlined version because it's going to go back to back with the NetRF meeting. So the idea is it's going to be a 1 day meeting with a maximum of 50 attendees. So we'll put out a call for kind of research summaries, and then we'll kind of have to go through. And if there are more than 50, we can sort of work out how we're going to do that. But it'll be based on groups and sort of research. It's going to be once again free. The Simon Robinson Prize is going to be awarded again, and the 4 subgroups are going to be maintained. But this one, because it's only a 1 day meeting and is going to be streamlined, there will be no white paper that comes out of this. Plus, I think it would be ambitious to get one out every single year. So it's going to be just research talks. And then the idea is that we'd move back to the UK or Europe in 2027, where then a white paper and a workshop would be included as part of a 2 day meeting and it would be standalone. Um, but just finally to kind of say thank you very much to all the kind of subgroup leads that have really helped get this off the ground. Also to both the NetCats Foundation for helping to support this meeting and the future meetings, and also to NetRF who are helping us and supporting us going forward as well. I'd also like to say I have spoken to a few other support groups who are also interested in getting on board with this going forward. So hopefully those names and logos will increase as the years go by. So thank you very much. That is the end of that presentation. So any questions, please feel free.
This is so exciting, Kate. Thank you for, really pioneering this and making this happen.
Well, I have to say, I didn't do it alone. There were lots of people involved over the years. So it's lovely that there are some enthusiastic people as well that really have stuck with this because we've had some ups and downs. And I think we weren't entirely sure that it was going to continue. And I think what I'm really pleased now is that now we've got some foundations involved, that it becomes bigger than me and Rachel and Poe, in that hopefully we can step down at some point and hand the reins to a younger, fresher person who's going to continue this for years and years to come and evolve it and make it bigger as time goes on.
Absolutely. Yeah. Sunita, I think you're muted.
I,
And
yeah,
Karen,
go, John.
So I just want to give a little bit of update. After I come back from UK, and we, with the MD Anderson strong support, we apply for the provision patent, and which is complete. And within the year, we will get the real patent. Yeah. So
Congratulations.
we talked to the MD Anderson. They are very interested. We complete. It's like because we have 1 year and the probation time or whatever since the first presentation. So after we come back, I thought we might can apply. At the first beginning, Dr. Yao said we might. Probably MD Anderson has so many advancements, they probably not interested. And I talked to them, I emailed them. They immediately said they are interested in the support. Within 2 weeks, with all MD Anderson strong lawyer team, we complete all processing. This is the one update. Another one, I think Kate just said, just said we present so many place for our models. You guys still didn't see official publication because I don't know, we want shooting high, but we don't know the results. And we do get, very good story right now, and we are shopping around, and hopefully we can, you guys can see our paper before the next meeting.
That would be great. We will look forward to that.
And also after we publish, we're gonna share our resource. That's what Dr. Yao said, our leader. Yeah, we're gonna share it with the whole community.
Yeah, that would be great. Thank you.
very exciting.
Those are great updates. John, you want to go ahead, and then after John, we'll go back to Sunita. I think she had a question.
Oh no, I just unmuted myself because I was going to ask a question that Zhang Jing addressed, and then also note that she was muted.
Are you already in MD Anderson now, John?
No.
Yeah, John, do you have an update for everyone?
I do have an update, but not.
You told me you got an offer as scientist or whatever.
I got an offer
and then you deny.
At Cleveland Clinic.
Oh.
Yeah. Well, we should talk. I think MD Anderson would be a great place to be. But don't tell the people at Cleveland Clinic right now because I just started like
No,
six
no,
weeks
no, no, no. No, that's
ago.
your job. That's your job to
Give me a
distribute
few
this all.
years.
Yeah.
Yeah, no, I just started at the Cleveland Clinic at as a project staff, which is I think kind of similar to a research assistant professor. And then I'm going to get appointed as an assistant
Oh, that's
professor,
good. Yeah,
the
that's
case
good.
president.
That's good
Yeah, which is
position. Yeah.
fun. Yeah, it's very good, very exciting. So I've been there for like 6 weeks. I think that'll really help me and help us, and there'll be really good opportunities that come out of that.
Yes,
Thanks.
congrats, John.
Thanks.
Congrats.
Sunita, did you have a question?
Yes. So, Kate, just something to think about. So, we call it the NET Models Consortium. Now, NET is not the disease of just one tissue, unlike colon cancer, breast cancer. So, how do we unite all these investigators who work on their favorite NET? You mentioned AI. If there's somebody who is an AI whiz, can they look at all of the data out there for all the different tumors, at least the RNA sequencing, and come up with something that unites all these different nets? Is there something that, something to excite everybody? Say, okay, here is a set of genes that is present up or down in all the nets. Can we unify to bring it under an umbrella and see how we can work together in that way? Something to bring people together. I think it's still pretty scattered. Lung, pancreas, gut, what have you, and very, very few investigators working on these different nets. So got to think of something, how to unite the tumors and how to unite the investigators. Something to think about. That's it.
Yeah,
And
well, I think that's
patient
kind of,
groups.
oh,
I just
no,
want
go
to
on,
say
John.
and patient groups. It's not bring everybody together.
Well, I think that's it. And I suppose the big question is there a thing that unites them, or should they be being researched separately? And if the latter is true, we need more people to be interested in researching them. Because I think what we're discovering is that, like, I had a conversation earlier with the guys at the clinical side in Oxford, and they're sort of saying, well, there aren't that many people to work with because there aren't that many people. So, you might be able to get samples from all these different things, but is there someone locally who's interested in lung nets and someone else who's interested in pancreatic nets and someone else who's interested in the pituitary? And the answer to that is no. And I think, I mean, I know for me personally, my bandwidth, my brain can't handle like all of those things simultaneously. So, if someone could find a thing that would unite, so we could put some big grant together to sell it to someone to say, this would be really good to do, then I mean, I'm up for that. I'd have no idea how to use AI to do it or whether I'd believe it if it did do it. But then I think what is really important to us is to get bodies in a room to discuss these things. And I think almost weirdly, it's got to be pushed by the patients to some extent as well. Because I know, like in the UK, you have sort of centres of excellence and they kind of congregate together in certain places. And it's whether, if you have a really good lung surgeon who's seeing all these things, who can like then refer it back and you have those connections. And I presented this actually at the UKNets meeting, which is purely pretty much a clinical meeting. And I think we were saying we have to shout a bit louder so the clinicians know where we are and what we're doing, so that when they do come across this stuff, they sort of go, oh, hang on, isn't there a person here that might? And I think that's what we've been really bad at. So I think we were saying that if there is something that unites us biologically, great. But otherwise, I think we all just need to shout a bit louder so that people know where we are. And then hopefully we can encourage more people to get involved and it just kind of snowballs from there.
I want to follow up on that. Well, I had a lot of good thoughts. Maybe we can talk. I think they're good. I had a lot of thoughts that I want to share. That's what I really meant. Um, uh, Maybe later, but it sounds like what you're really talking about there. I think I'm starting to understand a little bit why one might think of about a website or whatnot. Is you're talking about building a community, but you have to kind of build a community without the funding to begin with, and get people excited and interested. And then there are some other more rare diseases that you could probably look towards. It's a chicken and an egg thing. I know that at Amend USA, we've been in a place where it's just hard to get started. It's like it's a small business basically. And so we don't have much funding, which means we keep not having much funding, which means we keep not having much funding. And then building out that network has taken a long time. This group has been a big part of building out that network. Thank you. And I think we're getting closer. So maybe there's some other communities to look at that could help with that, building out that network and that excitement. Um, but I see. I guess what I really wanted to say is that yeah, that I think that really that makes a lot of sense. That's a real key need so that because I don't think anybody's ever going to be all together, especially in the United States. And so you just have to uh, make sure that if you will, that the person at Vanderbilt knows that their vendor of choice for this particular kind of thing is at U Chicago, and so that they're staying connected. Um, so how do we do that?
Yeah, I think that's been a big discussion topic for us that we all have our own expertise and there's no point in all of us trying to be an expert in everything when there are people sat there. And so it's how do we find out who those people are and where they are? Because I think a lot of them, especially on the science side, are kind of doing things kind of as a sideline. So they
Mm,
might be including nets as part of, they might be a lung cancer researcher and
mm,
they're like, oh, well, I'm mainly interested in another type, but oh, I happen to have these nets. So I'm kind of bundling it in with everything else I'm doing.
mm.
And I suppose it's kind of a bit like when that stuff rears its head, and actually the patients and the clinicians are kind of the easiest inlet to kind of also making that noise. Because if you're diagnosed with a lung net and you've gone to the patient advocacy group and gone, hey, hang on, what's going on here? Like you can speak to the clinician who can go, oh, well, I wonder if there's a researcher, because it's going to be quite hard for the research community. I mean, we'll try, really, really, we
Yeah.
will try to get everyone on board and we will keep presenting and standing up and saying things. But it's very hard to infiltrate some of these really big networks and meetings to kind of
I,
get
I
heard
like
in
I
amongst
like
all the
where
other
your
noise.
thoughts are going, Kate. And I think what I'm hearing is start inviting the expert of every net type and bring them together in the room and have a
Mm.
discussion as to how can we all unite and work for this cause. Yeah, very nice, Kate. Thank you for your thoughts.
So,
Thank
from
you.
a patient's perspective, are we registered with Orphanet and also with Eurodis so that we're with the rare disease kind of community that um, mm, is seen? And I liken it. I was a patient in the hospital a couple of years ago, and there's always a feedback survey. So I filled out the feedback survey and I said, I'd really like to see that rare disease is the raising of awareness for rare disease in throughout the hospital. And of course, we've got Rare Disease Day on the 28th of February. So out of the blue, I happened to be in the hospital this week, and here's this great big wall poster, International Rare Disease Day. So I was absolutely thrilled that they had moved forward and they were showing something, and I'm thinking, good, this is what we need. Um, and it is hard to bring all the different disciplines together. I belong to so many groups, only because there is no um, centre of excellence here in New Zealand. Um, they very close in Canada to having that, and yeah, that's just me. I just get on the bandwagon sometimes and I get excited.
No,
Well,
that's
Thank you for
awesome, Tanya.
sharing. Yeah.
Yeah,
we're
and that's
happy
the
to have you.
example of using your voice,
Yeah,
you gave that feedback and they listened. And that's a
Yeah,
great example.
that's what we need more of. We need more patients getting on the bandwagon and making
Yeah.
their voices heard.
And I think that's not to be underestimated. I think that does make a bigger impact than patients realize. And I think also something else you sort of alluded to is breaking the barriers of this international situation, because I think there are a lot of national organizations. And I think what we're trying to do is kind of see if we can bridge that gap that you might have your local support group in whichever country you're in, but then are you finding out about opportunities internationally, or are they
Mm,
more centric around what's happening locally? And I think that is kind of something we wanted this consortium to sort of think about, that it's breaking down those international barriers, of which there are quite a lot, because there are so few of us in each field. That is going to be so important that, the lung people from all around the world can come together, and then there might be five of you as opposed to one, but you're not going to do that within one country probably. And yeah, the more patients we have getting involved and looking at that stuff, great.
So you've probably heard of ComNets, the Commonwealth Neuroendocrine Tumor Group.
I
So that
have. That's
I
probably
have
because
not.
I'm Canadian.
yeah, so it's Canada, Australia, and Canada, Australia, and New Zealand.
Yep.
But Commonwealth is Commonwealth, so Britain should be in there. Hello.
This is what I mean, and it's like, we're
Yeah.
not. We're not going anywhere.
No, I've never heard of that before.
No.
ComNets? No.
No, I haven't. I'm going to do some research.
Simon Singh out of Ottawa,
Toronto. You
Toronto,
Toronto.
Sunnybrook.
I mean, if it's Commonwealth, I don't understand why the UK is not in there.
That's
Because you
weird.
guys excluded us to be like the colonies, and yeah, so that's why we have to be like by ourselves. It's like part of the Commonwealth only.
How should that's my job for tomorrow. Unbelievable.
Yeah, there's lots of researchers and scientists in that group.
Okay,
Yep.
I'm going to be
So I
busy
see
tomorrow.
myself as a patient expert because I attended the summer school in Barcelona with Eurodis. And yeah, that's a term that's not commonly used, certainly in New Zealand, but in Canada, where I was living before, it was recognized. I'm a patient expert in my condition, not not everything. Yeah.
See you,
Yeah,
Sania.
no, it's okay. I guess yeah, it's getting late, but
It is.
I just wanted to say that I really think it's a great idea that you're thinking about like having a website and to kind of consolidate the resource and all that, and also like what Kate mentioned about the guidelines, like for example, like just like the taking the exosomes group as an example. I think that that's a great idea because last year, I think around this time, there was like a prestigious like Cancer Cell or something like paper where, then they didn't characterize their PDX model and clearly I think it lost a lot of the net feature because their tumors were like this big, like 2 centimeter in 55 days. And so we've all worked with that. And it's sort of like we know that that's needs further characterization, and people can't really just say that they made these model, and it affects everybody's work and everything too, because then people are like we're trying to put a lot of effort to developing these hard and difficult to develop models, and then other people are just like, oh well, we have them, but they haven't fully characterized their models. So I think if we have like a minimal guideline that okay, well, you need to look at least the four markers like SSTR2, somatostatin, well, I mean synaptophysin, chromogranin A, and all these like basic stuff that a lot of people unfortunately don't know, and so I think it's unfortunate, but I think it's something that needs to be done. So I think
So
it's
well
thank you
volunteered
for
for
being.
doing that, Poe.
I have an
I
idea on
think,
that.
to be fair,
And
we did say what we thought would probably be useful, and I know this was a big discussion topic at the meeting, which is not a small undertaking, is to do a systematic review of the publications that exist, how many of them would meet the guideline criteria that we're going to write, and sort of then say, because what we basically want to do is, like, I know people are applying for grants and then they say, well, why are you doing that? There one exists. And
Yeah,
it would
or
be
it's
really
been done
nice.
before, but that's the most frustrating part. But they're like, why are you even trying to do this? It's been done. Look at this awesome paper. But then they didn't do the job. So, and we're like, we still don't have models to study our patient tumors and how they behave and which drugs actually work really well for subclasses and all that, but or some of us have been like doing really extensive work to just get there, and then other people just come along and say like, we got them, it's so easy, which is not really true. Yeah.
Yeah, I mean, I think there is a big need for that. I think it's just the time
I, mhm.
and effort to do it that I think, doing a systematic review and then kind of a consensus guidelines. I mean, I do wonder whether that's something that we might want to address at one of these meetings, whether we have a working group that's put together or like who gets together and just says like, right, they're going to write that between them. And then it gets consulted. I was involved like in the MEN1 guidelines that came out, and that's kind of what they did, that they sent surveys out to pretty much all the experts and said, what do you think on this? And then they were all listed. Every expert that was contacted was listed sort of as an author on the paper in like a sort of consortium sense. But then there are a few people that sort of headed it up. But it's like there are ways and means you can do it to kind of cover a lot of bases and get all the experts, which gives it a bit more clout. But it's, yeah, going to take time to do something of that detail. So it's how quickly we want it out. And then, because I know, for example, the International Journal of Cancer is very hot on cell line authentication, and they were the ones that spearheaded that. So there was talk about approaching them for a publication like this because it's kind of within their remit. So they might be interested, and whether it's worth contacting them to see kind of what their expectations would be, and whether they would, in theory, take something like that. But it's the workload that is entailed, I think, is the biggest challenge with all of it.
At the
Go ahead,
upcoming
Rachel.
at that thanks at that upcoming meeting, maybe these are things that you can put on the agenda and try to decide, well, what do we think, what's the lowest hanging fruit versus the biggest bang for the buck, and then get everybody around that. Maybe there isn't a common biology, but there's some other common need that everybody can come around to.
Yeah.
Yeah. So I think we have a lot of exciting work in our future. But we're 11 minutes past the hour. So I'm going to go ahead and wrap up this conversation, even though it's excellent. And we have a lot more to talk about in the future. So we'll see you guys next time. Selena, do you have anything to say? And a big thank you to Kate. Thank you so
Yeah,
much for
thank you.
presenting.
Yeah, thank you, Kate. It was a fantastic meeting. It was great to catch up with all the things that have gone on. I'm really looking forward to the white paper and looking forward to the next net model meeting. So anyway, all I've got left to say is the next meeting is on the March the 26th. So yeah, very exciting, and hope to see you all there. So thank you very much again, Kate. Thank you everyone for coming. And I'm going to be really busy tomorrow with this Comnets. If anyone has any news or updates or knows anything about it, then feel free to email me. So any more research would be great, and I'll have a look at that and see where we can go with that one. So, but thank you very much, and I'll see you all next month. So thank you.
Bye,
Thanks a
everyone.
lot. Thanks. Bye bye.
Bye,
Bye
everyone.
bye.